Ever wonder how a copy of our DNA ended up in every cell in our bodies? Well, this process starts with a process known as DNA replication. In order for replication to begin, the replication machinery must have access to the nucleotides that are encoded in the DNA strands. This access is granted through the enzyme known as DNA helicase. Imagine for a minute that you are shopping and interested in a new pair of jeans. You want to try the jeans on, so you need to figure out how to get into them. The first thing you notice is the button. Opening the button gives a little room, but not enough for you to get into the jeans. Then you pull down the zipper, which opens the jeans up even more, giving you the space you need to get into them. This is very similar to the role that DNA helicase plays in DNA replication. DNA helicase is the zipper. It unzips our DNA strands to allow space for attachment and to expose the nucleotides that are used as a template during DNA replication and transcription. Let's recap a few things about the structure of DNA so we can better understand why DNA helicase is needed. Remember that our genes are composed of two DNA strands that are wrapped around each other to form what is known as a double helix. The two strands contain complementary nucleotide bases that are bound to each other through hydrogen bonds. Usually, hydrogen bonds are pretty weak bonds, but in the case of DNA, they are very important and not easily broken. That is, until they meet DNA helicase. In order for DNA polymerase, in replication, or RNA polymerase, in transcription, to see the nucleotide bases that are to be used as a template, these hydrogen bonds must be broken to expose the much-needed nucleotide bases. DNA helicase causes the hydrogen bonds to melt, which opens up the DNA at the site where DNA helicase attaches to the DNA double helix. This action will continue as DNA helicase moves down the length of the DNA strands, just like a zipper gives more access the further down it's pulled. As DNA helicase continues to move, it creates a replication fork, which is the open area of DNA where replication or transcription can take place. Unlike zippers on genes, DNA helicases do not work alone. Our DNA is pretty lengthy, so this process would take forever if only one helicase were working at a time. For this reason, there are multiple DNA helicases at work on the same strands of DNA, but in different places along the DNA. The different areas of open DNA are known as replication bubbles. You should now understand that DNA helicase has a very important job to do. It is responsible for opening up our DNA to allow for replication as well as transcription of our DNA. A DNA helicase is an enzyme that functions by melting the hydrogen bonds that hold the DNA into the double helix structure. The area of the DNA where the DNA helicase has unzipped the DNA is known as a replication fork. Multiple DNA helicases work along varying parts of the DNA, creating replication bubbles along the DNA strands.